Autophagy plays a powerful neuroprotective role by preserving the delicate internal balance of neurons, which are among the most energy-demanding and long-lived cells in the body. In the brain, the maintenance of cellular integrity is critical not only for day-to-day function, like memory, attention, and coordination, but also for preventing the cumulative damage that leads to cognitive decline. Autophagy is the brain's internal recycling system, identifying and breaking down damaged proteins, dysfunctional mitochondria, and other debris before they can disrupt neuronal communication or trigger inflammation. As we age, the gradual weakening of this process becomes a major contributor to neurodegenerative diseases. Enhancing autophagy is increasingly recognized as a central strategy for slowing cognitive aging and protecting against disorders like Alzheimer's. One of the most toxic threats to neurons during aging is the accumulation of misfolded proteins, particularly beta amyloid and tau. In healthy neurons, autophagy helps eliminate these proteins before they reach pathological levels. However, with age, this clearance process becomes less efficient, allowing beta amyloid plaques and tau tangles to accumulate in brain tissue. These aggregates interfere with synaptic signaling and initiate inflammatory responses that further degrade neural circuits. The result is a progressive loss of cognitive function, beginning with mild memory lapses and eventually leading to severe dementia. By restoring autophagic flux, the continuous formation, maturation, and degradation of autophagosomes, the brain can regain its ability to clear these toxic proteins, preserving neural networks and supporting mental sharpness. Beyond protein clearance, autophagy also maintains mitochondrial health in neurons through mitophagy. Neurons require large amounts of energy to sustain constant electrical activity, and this energy comes almost entirely from mitochondria. Damaged mitochondria not only produce less energy but also leak reactive oxygen species, damaging surrounding cellular structures and DNA. When mitophagy is active, these faulty mitochondria are removed and replaced with new, functional ones, maintaining the energetic and metabolic balance that neurons rely on. This is especially important in aging brains, where mitochondrial damage tends to accumulate. Supporting mitophagy through lifestyle or pharmacological means has been shown to enhance brain plasticity and resilience, delaying cognitive decline. Autophagy also contributes to synaptic remodeling and plasticity, the brain's ability to form new connections in response to learning, experience, or injury. During development, and even in adulthood, autophagy helps shape and prune synapses, refining neural networks to be more efficient and adaptive. This process is essential for learning and memory, and its decline with age may be one reason why older individuals often struggle with acquiring new skills or processing information quickly. By keeping the synaptic environment clean and responsive, autophagy enhances the brain's ability to adapt, reinforcing its cognitive flexibility well into later years. What makes this even more encouraging is that autophagy can be stimulated through relatively accessible interventions. Fasting and calorie restriction are perhaps the most well-documented strategies. Both lower insulin and activate AMPK and SIRT1, key regulators of autophagy in neurons. Exercise, particularly aerobic activity, not only improves blood flow to the brain but also enhances autophagic activity in hippocampal cells, supporting memory and spatial navigation. Nutrients such as omega-3 fatty acids, curcumin, and compounds found in green tea, like EGSG, have shown the ability to stimulate neuronal autophagy and reduce beta amyloid accumulation in animal models. Sleep also plays a vital role, as deep, slow-wave sleep is associated with increased clearance of brain waste through both autophagic and glymphatic pathways. Importantly, some emerging therapies are now targeting autophagy more directly in the context of neurodegenerative disease. Drugs and natural compounds that mimic caloric restriction, activate autophagy-related genes, or inhibit mTOR signaling are being explored as potential treatments for Alzheimer's, Parkinson's, and other forms of dementia. While many of these interventions are still in early stages, their effectiveness underscores how central autophagy is to maintaining cognitive vitality. In summary, autophagy acts as a silent guardian of the aging brain, removing the internal threats that compromise memory, learning, and mental clarity. By preventing the buildup of toxic proteins, preserving mitochondrial function, and maintaining synaptic plasticity, autophagy gives neurons the tools they need to endure over time. Unlike many aspects of brain aging that are difficult to reverse, autophagy remains inducible, even in older adults. This offers a remarkably hopeful message. Cognitive aging is not solely a story of decline, but one of potential renewal. Through intentional habits, supportive nutrition, and future therapeutics, we can harness the body's own recycling system to protect and preserve the most essential organ of all, the mind.